So Herschel's right. You know, this whole idea of these harmful algal blooms has been politicized significantly. You know, it occurred to me as I was thinking through how I wanted to frame this, that it seems like many times we have lost our way. And what I mean by that is that we should be driven by the truth. We should be driven in a pursuit for legitimate truth, right? I know that in the political realm today, it's easy to simply just make an accusation or a comment. I'm not talking about Judge Kavanaugh. I'm talking about politics as a whole, because it impacts every life here and every life that is in contact with every life here. So we should be thoughtful individuals about truth. We should seek truth. And most importantly, we should protect ourselves from half-truths, quarter truths, incremental truths, not whole truths. We should protect ourselves. When you read something, when you hear something, especially as it relates to the environment here in Florida and especially as it relates to this topic, I would encourage you to make sure that you reset your brain and you do not allow yourself to get sucked into this ridiculous thing that is going on today in our society. A lack of truth. Truth's all we can stand on and for. Now, what I will tell you is these folks have real expertise. They are the real deal. And they're going to speak truth. And what I want to say is that you're going to be able to believe the things they say. So Luna Phillips, down on the end, is a lawyer with Gunster, leads the firm's environmental practice. She has in her background been the senior attorney for the DEP and SwiftMud. Dr. Kate Hubbard. I'll just read the first, you'll understand what I mean when I say it. I'll read the first paragraph. Dr. Hubbard's research on toxic algae incorporates traditional and molecular ecology and taxonomy to consider the varied abiotic and biotic mechanisms that influence the biodiversity, distribution, toxicity, and evolution of these organisms in the marine environment. She's well qualified. Let's see. And then Ed, I must apologize, and with the last name Albritton, it gets butchered a lot. Would you help? Legere. That's cool. Ed Legere, founder and CEO of Algal. It's a biotechnology company that essentially grows algae for profit. What an interesting concept, right? And then Dr. Barry Rosen worked with, and here's a qualification, has worked on cyanobacteria for over 4 decades. I think that says enough. Okay, so as we get started here, anybody want to make any kind of brief opening statement? No? Wow, how about that? Cool. So everybody, all right, sir. So how about we do it like this? I'm going to pitch out a question, all right? And then you can chime in, because there are just a couple of topics we want to be sure and touch. So here's one, fish kills. You've seen the pictures, right? Fish kills. Is it related to red tide, green tide, both other factors? Can you give us, or would you give us some perspective on how we as the public need to be understanding?
This is on?
Yes.
Okay. Wave in the back if you can't hear. So in terms of the fish kills, I will touch a little bit upon this in my talk, but the red tide began in November. Since then, we've had over 1,000 reports to the FWC fish kill hotline. There have been other wildlife impacts, manatees, sea turtles, and these have all been related to red tide. There have been relatively few fish kills that have been associated with this year's cyanobacteria bloom, and those that have been identified were thought to be due to low dissolved oxygen rather than any impacts from the toxins themselves. And maybe Barry wants to comment a little bit more.
Yeah, that's exactly right. When you get this big biomass, big glob of blue-green algae, it's the oxygen depletion because there's bacteria there and the cells are dying. That's what causes fish kills 99% of the time. Fish are really resistant to the actual toxin that they make. They have great livers that can detoxify. There's very rarely do we find cases the toxins from cyanobacteria actually cause a fish kill. Wildlife is a different story, but fish themselves are pretty resistant.
All right, good deal. All right, so I've been given some direction. So, Luna, you'll get started. You'll do some commenting for us and give us some direction, and then we'll move down the way. How about that?
Thank
That
you,
okay?
Representative. We're actually going to start with Mr. Rosen. He's going to do his presentation first.
Okay.
Then Dr. Hubbard, then Mr. Legere, and then what we thought is open it up to discussion from the audience, and we can have a more interactive discussion after you see their presentations. if
Well, that's
okay.
cool with me.
Thank you.
All
Little
right.
did you know.
How about that? Yeah, can I sit here?
You can sit right there. That's the seat of honor.
There's a few phones for you to use.
Can we turn the lights down a little bit? A little mood for cyanobacteria. There he's going. Okay, so I'm Barry Rosen. I work for USGS. Before that, I was Fish and Wildlife. Before that, NRCS. Before that, South Florida Water Management District. And once upon a time, a faculty over at the University of Tampa. And almost in all those positions, I worked on cyanobacteria, drinking water for the city of Tampa as well as Sarasota, taste and odors, that kind of stuff. All right.
Right here.
So, I'm really bad at telling jokes, so I wrote this one down. I am a phycologist. And phycos comes from the Greek word algaeologist. You all know what ologists are, so a phycologist. For the longest time, my parents thought I was a psychologist. And I had to explain, yes, I'm doing the psychology of algae. And that's true. As an algal physiologist, I try to understand what they're doing, when they're doing it, and why they're doing it. To kick off, algae is such a broad group. And this is the family tree. And I want to point out, this is anything that's a colored box is an algae. And if we look at cyanobacteria, they come off the tree very early. They are truly bacteria. And if you look for for dinoflagellates, they're way up here. Animals are just this little branch over here, right there. There's animals and land plants. So the diversity that we call algae, there's one thing that unites all algae, and that's that they have chlorophyll A. Even cyanobacteria have chlorophyll A. There are other chlorophylls too, but chlorophyll A is what unites all the algae together. And when I learned phycology back a million years ago, guess what? We took the blue-green algae, and that's what we called them. Only recently are we calling them cyanobacteria. Well, they're bacteria, like I said, and they've got the cell wall of a bacteria. And if you remember back to your biology, in high school or college, prokaryotic organisms have no nucleus. They have no mitochondria. They have no internal bound organelles. If I get too deep into the science, you can groan. If you're moaning, then you got the joke. So anyway, but they have membranes that hold the pigments that I'm going to talk about briefly. They're gram-negative bacteria. Enough, I don't want to go too deep. So what's important is they do have chlorophyll A, it's down here, and they have a compound out here called phycocyanin. And if you hear cyanin, that's the color, that blue that you see. Oftentimes when they die out, it's from phycocyanin leaking out of the cells. It's part of an array, an antenna that captures sunlight that they have to channel and funnel down to chlorophyll A for photosynthesis to happen. And remember, photosynthesis is really important. That's how oxygen came on the Earth. And the cyanobacteria actually oxygenated the Earth initially. And that's how all the higher organisms evolved, because of oxygen. So you capture, you split water when you do that and you make oxygen, but you also capture energy, making ATP, things like that. So they have the thylakoids for capturing that sunlight, and that's really important. So blooms look like this. If you were to look at it with your naked eye, you can see this is a microcystis bloom from this past summer. It looks like this, and you hopefully don't get it really thick like this over here. This is Lake Okeechobee, an aerial photograph. And when it dies, here's that blue leaking out. If the lights were a little lower, you could see this better. This is what is going on. It leaks out when the cells die. The toxin they make is also inside the cells. It only leaks out when the cells die. Now, if you eat the cells, of course, you're going to be exposed to the toxin. So one thing to note is that all of this stuff is floating. And that's the next thing I'm going to talk about. So besides having the pigments and being able to capture sunlight and do photosynthesis and grow, why are they so successful? Why do they form blooms? It's because they float up to the surface. And they float up because, again, no membrane-bound organelles, but this big structure here is a bunch of hexagonal gas vesicles that actually hold air, and they get light and they float up in the morning. They photosynthesize. What they make makes them heavy. They sink. So they go up and down in a diurnal pattern. And when they make that compound, they're making a carbohydrate, and they sink. When they go down in the water column to get close to the sediments, and that's where phosphorus is often released from anoxic sediments. They pick up a load of phosphorus, come back up and down, day in and day out, until something disrupts that balance. They lose a key nutrient, and they can't regulate their position in the water column. That buoyancy regulation is not unique to this kind of bacteria, but certainly the cyanobacteria have that advantage. Not all blooms are at the surface, but the ones that we're talking about today are surface-forming blooms. And then you get this, the green slime. This is a Calusahatchee. This is from a few years ago. And why do we really care? Well, you've got the toxicity issue. But again, I talked initially about how the dissolved oxygen sag, when you've got this much in the water, in the day it's photosynthesizing, making oxygen. It's also using and burning sugar. You have to do that. But at night, that's all they're doing. And when you respire, you give off and you utilize all the oxygen in the water, causes that dissolved oxygen sag, and the fish that can't get away from it, if it's a very extensive bloom, die. Taste and odors were a long-standing issue for drinking water facilities, and of course aesthetics, unless you like the color green. No moans. Okay, I'm going to only talk about a couple of the traits, just about 24 traits that make cyanobacteria, blue-green algae, so successful. The second one is a key element of any living organism inside of proteins is the element nitrogen. You need phosphorus, but you need even more. You need 17 times more nitrogen than phosphorus. If you run out of nitrogen in a water body, certain cyanobacteria aren't going to make it. Microcystis is going to crap out. It needs nitrogen. It needs phosphorus too. But there's a whole group of cyanobacteria that have a specialized cell. This cell right here. It looks like this right here, and it looks like that. And in that cell, it is able to take atmospheric nitrogen, N2. 78% of the air we breathe is nitrogen. And they can incorporate it and make proteins. Skip all the detailed, the chemical steps in between. But because they can do that, it gives them a tremendous advantage over the non-nitrogen fixing organisms. The bacteria in the roots of alfalfa and things like that can do it too. It's one of the few organisms on the earth that can fix atmospheric nitrogen. Huge advantage. So if you run out of nitrogen, and this happens in Lake Okeechobee, and there's a bunch of cyanobacteria in that lake too, the big bloom in '86, I was not around, but shortly thereafter, that spawned the Lake Okeechobee and the whole swim program in Florida was because of a nitrogen-fixing cyanobacteria in Lake Okeechobee. 42% of the lake was covered with it. So you've got these organisms, they've got their specialized cell that fixes nitrogen. It passes that nitrogen on to adjacent cells. The term for the cell is a heterocyte, as if you want to know that. But there they are. They're not that abundant, but it's in there and it feeds all the cells next to it. So if you've got it, you've got a tremendous advantage. The organism Microcystis does not make nitrogen. It has to obtain it from the water. And that's the one that dominated this year by far. I barely saw any of these others this year. One or two other quick things, they're bacteria. The warmer it is, the faster they grow. You can have an organism that starts out at one cell in the morning, you get a division a few hours later, another one. So one cell can become 8 cells in one day. So temperature is important. Temperature hasn't really cooled off that much in the water body, but the cyanobacteria bloom is kind of gone in Lake Okeechobee. You have to wonder why. So it's not simply nutrients. There's plenty of nutrients out there. It's not simply temperature. There's other factors we just don't know that cause these blooms and maintain them. I think it's day length that has part to do with it. We're starting to explore that. So I'll skip this. This happened in the Hillsborough River, temperature and the bloom. And so we care about this stuff because there are cattle deaths that die. That fish, who knows exactly why it died, but cattle death is frequent, especially out west. There's a bunch of toxins. You've got liver toxins, nerve toxins, skin toxins. You pretty much have to ingest it other than the skin toxins. The new one on the scene is BMAA. It's like an amino acid that we use. You can accidentally incorporate it and you cause protein damage. And maybe it might be linked to things like ALS and other things. These are potent toxins. The most potent toxin from cyanobacteria, saxitoxin, is at 9. This is LD 50, 50% of the animals die. There's racine, the most potent toxin. There's cobra toxin at 20. Look at some of these algal toxins are way up here. Microcystin is one we care about most right there. Okay, I'll skip the chemistry, but we call this stuff fast death factor. It's a liver toxin. The microcystin is the most common. Basically, why is it a liver toxin? Here's a normal healthy capillary over here. This is what happens when you ingest microcystin or the organism Microcystis. The liver, the capillaries pull apart and it causes a bloody liver and death. There are regulations. Well, I'll take that back. There are guidelines from EPA in drinking water. And so there's concentrations that you have to obtain, or if you're a drinking water facility, it's 0.3 micrograms per liter and 0.7 for another compound called cylindrospermopsin. And it's a 10-day exposure for children that are a certain age. It's a little convoluted, but we have guidelines. What's the most important part of this picture? Beer. Oh my God, you got it fast. So really the point is that it's not just drinking water. This stuff can get into fish through zooplankton. It can get into the mussels and clams on the freshwater side. So it's important to think about. All right, I'm wrapping up. So when you go to a water body, you have to look for the signage. This one says, no lifeguard bacteriologist. I had to add, of course, phycologist in here on duty, so swim at your own risk. And cyanobacteria are special because they're artistic, and I take a lot of pictures. Thank you. And if you need more information from me or about the work I'm doing, it's okay to say this, I hope. Just Google me. Use Barry Rosen cyanobacteria. I did it this morning, and a bunch of stuff cropped up. Thank you.
Having all kind of trouble. I'll swap with you. How about that?
Great.
Hi, everyone. Thank you, Barry, for giving a great introduction to algae, and I'll try to skip through. Just to give you a little background about myself, I started working on harmful algae as a student at New College in the 90s. I went away and did my graduate work on the West Coast, did my postdoctoral work on the East Coast and the Northeast, both places working on harmful algae and those systems. And so now I've been very excited to come back to Florida, and I lead the harmful algal bloom program for the Florida Fish and Wildlife Conservation Commission. I'm also a guest investigator at the Woods Hole Oceanographic Institution. So just to give you a little bit of background, and also I wanted to mention my bio is probably the most complicated part of what you'll hear from me today. But it's really important to remember that we're hearing a lot about algae today, and algae are really important. They form the base of the food web, and so we have to remember that, and only a small subset of algae actually are capable of producing toxins or being harmful. All algae require similar things. We heard a lot about that from Barry, light, nutrients. There are also specific environmental conditions that algae need to be able to grow. And in particular, when we have harmful algal blooms, that's usually a proliferation of cells, and it can cause a bunch of negative impacts. And I'll show you some of those. Okay, so first I wanted to just give a quick overview of red tide. This is a dinoflagellate. I'll show you a picture in just a minute. It produces a suite of toxins known as neurotoxins, brevetoxins. These can kill fish, birds, sea turtles, manatees, dolphins, other marine mammals. They also can cause a problem when filter feeding organisms like shellfish concentrate the toxins that are in these algae. That can cause neurotoxic shellfish poisoning, and so we very carefully monitor our shellfish beds working with the Florida Department of Agriculture and Consumer Services. The toxin can be carried as well as being ingested by bivalves, which can present a threat to human health. It can be aerosolized in beach spray, and that can cause respiratory irritation problems for people, and it can cause severe problems for people that may already have impaired health. And then you can see some of the images here. These blooms can cause massive fish kills on beaches. They can cause problems for local communities that have a lot of tourism that's very important to them, and the costs of cleaning something like this up are really high. These are two images. Blooms do definitely stand out. These are both taken from aerial surveys, and you can see the individual organisms that are causing these different blooms. We have red tide over here, and blue-green algae or cyanobacteria. This is microcystis over here. This is one cell of Karenia brevis. These are basically colonies of microcystis. You can see that they cause different colors in the water. I won't go through these in detail, but one of the main differences, and I will discuss this a little bit, is that red tide blooms start offshore 10 to 40 miles, and they're brought onshore by currents. Once they're brought inshore, they can then proliferate and cause a whole suite of problems, and they don't really thrive in beyond a certain salinity, but they are quite tolerant. So this is Karenia brevis. I don't know if the video will play, but this is the organism. Is it possible? No? We'll move on. No? Okay, well, if you were to see these cells, they actually swim around. So these are the cells that form the dense blooms that discolor the water that I showed before. And they do have a unique swimming pattern that we can use to distinguish them from other cells. Oh. And for additional questions, you can Google Barry. Barry, you weren't supposed to put that slide in my talk. Okay, so I'll go on. Oh, there you can see him swimming around.
That one in the middle is kind of sick.
Yeah, he's kind of just hanging out there. So these cells actually have two flagella, and they help them both steer and propel them through the water. These organisms also move up and down in the water column, which allows them to access nutrients at depth and sun at the surface for growth. So in terms of where we're at this year, I wanted to give a historical context for the present red tide event. These are showing some of the longest documented red tides since our monitoring program at FWC began in 1953. You can see that the longest documented bloom lasted for approximately 30 months. We're not there yet, thank goodness. That was in the 90s. However, we just did, as we moved into October, enter our 12th month of the present bloom. So you can see, going back in time, we've had other severe blooms. The duration is just one indication of the severity of a red tide event. This is our most recent status map. We put these out twice a week on our website. And you can see here that we are now experiencing blooms in nearly every part of the state. We have one in Northwest Florida, Southwest Florida. This has been going on for a year. And then just within the past week, we've had an appearance of Crania brevis red tide on the East Coast. The last time that happened was in 2007, where we had blooms in all three areas of the state. And I guess I didn't mention that, but each one of those dots represents a sample that was collected. And so this is showing the monthly summary of all the samples that were processed by our monitoring program during September. So Crania brevis is a very ecologically flexible organism. That means that it can thrive or persist across a very broad temperature and salinity range. So unlike cyanobacteria, there's not a clear demonstration with Crania growth and temperature. And this is based both on observations where we can grow cells in the lab, but also our field observations. We also know that Crania brevis, or red tide, is capable of using a wide diversity of nutrients for growth. And these include a number of different sources. We have sources from estuaries and land. We have oceanic sources that are involved with the deep upwelling of nutrients to the surface. Once we have a bloom, we have a whole planktonic community that includes bacteria, viruses, and those nutrients are constantly recycling and regenerating. We have fish kills, which themselves can be a source of nutrients as they decompose. And then we have something where Barry mentioned the nitrogen-fixing bacteria, the algae that can take bacteria from the air and convert it into other forms. We have a marine form, and that can play a role in Crania brevis or red tide events as well. So it's quite a flexible organism, and that's part of why it's so prolific. This is a bloom, an image showing several different things I wanted to point out. So this is that nitrogen-fixing bacteria. We call it trichodesmium, that's its name. And you can see that it has a different color. Here it's showing up as the golden color. We also have Crania brevis in close association with this trichodesmium. This was taken during one of our aerial surveys in September. And so these patches here are the red tide. And this is the trichodesmium. So you can see that there are numerous sources, and we've had, in particular this year, quite a bit of iron dust deposition that can help trichodesmium grow so that its growth isn't limited by the availability of iron. And then, as I mentioned, that can then help draw down that atmospheric nitrogen and make it available for red tide in other forms. This is the hub of our group. Since November, we've processed over 10,000 samples for red tide using light microscopy. We are really interested in working with automated technologies that can help improve our ability to screen samples. Those might include genetic approaches. We have handheld sensors that we can take out in the field. We also have microscopes that we can take out in the field that can quantify Crania brevis based on its swimming. We also are really interested in using technology that can be placed in the field to get a better sense of what is going on with these cells. This is an example of one of those instruments. It's actually an imaging flow cytobot, and what this does is collect a water sample every 20 minutes, and it takes a picture of all of the different cells in that water sample. And so you can see here, these are all different Crania brevis cells. And this allows us to get a much better sense of what the cells are actually doing in the environment at the resolution that we need so that we can get a better handle on what really causes these to proliferate. We also use satellites to help us track blooms, and so you can see here now our cell count data is overlaid over a chlorophyll image. Barry mentioned that cyanobacteria have chlorophyll, so does red tide, and so we can sometimes pick up that image from space, and that helps us know where the bloom is. This is back in September. And I wanted to mention that because this is a product. We work very closely with USF, and this is one of their products. And so we've been doing aerial surveys as part of this bloom. This is one of our law enforcement planes that we've been going out. We've been able to do surveys up to 10 miles from shore. And again, just to give you some sense of the bloom and its patchy nature, these are images taken 1 mile, 5 miles, and 7 to 10 miles from shore. And again, this is in this area, so it's not even in the area where we have the highest concentrations of cells. This is one of the images. So again, this is chlorophyll, and I just want to draw your eyes down here south of the Keys. So this is our southwest bloom that's been going on since November. You can see a little trail of the chlorophyll that comes down around the Marquesas. We suspect that this may have been the signal, the chlorophyll signal that was related to that Karenia brevis being swept up the east coast. So that's something that we're obviously keeping an eye on. We have, so this is, and you can't really see the arrows very well, unfortunately, but there are videos online that demonstrate tidal activity and how much mixing goes on based on the physics and the currents in these areas, and I can tell you that it's very dynamic. So even in the course of the day, we have water moving in and out of these estuarine systems, and that again makes it tricky to track the red tide and also to follow some of the water quality conditions. And unfortunately, this isn't showing up very well, but we use those models. I was showing a salinity model on the prior slide. We use those models to help predict where red tide is going to go next. These are like weather forecasts, and for right now, we have 3.5-day forecasts. And again, they help us know where the bloom is likely to go. And this is an image from Reddington Beach in September, and it shows you, again, how patchy. These are both incredibly high cell concentrations, and you can see this is right up against the beach. So in terms of where we're at right now with the current bloom, we've had a bloom that started last year. It's stuck around through the time where we typically see new blooms initiate. I just wanted to give a sense of that process. I mentioned that they start offshore, and with that process, we have a combination of offshore currents as well as winds that are helping to bring cells from offshore into the nearshore environment where they're upwelled into the surface. Once they're upwelled into the surface, they can take advantage of the different conditions there that can help them grow. This is an instrument called a glider that we use. It's basically an underwater robot that moves up and down each day and collects different measurements. I'm showing here an image. This was from August. It was from August to September. We had approximately a 3-week deployment. Again, this was with USF. So what we're seeing here is the chlorophyll image at depth. We've seen some of the images at the surface, and what we're following here is as we're going offshore, it's getting deeper. As we go near shore, it's getting closer to the surface again. So we're doing some loop-de-loops. This is where the glider stopped. These patches of chlorophyll are what we hypothesize could be the seeds to a new bloom that may be coming in at depth. And when we were able to collect the glider, we were able to also collect water samples at depth, and we did confirm that Karenia brevis was occupying those high chlorophyll signatures. We also have a number of other types of forecasts that we can use to help people be aware of what's going on, and also beach conditions reporting systems. And these are tools that, again, the public can use to help decide whether or not to go to a particular beach during a day. We have the beach conditions reporting system, which is operated by Mote Marine Lab, and then NOAA issues respiratory irritation forecasts twice weekly. And our cell count data is transferred to these different sources each day to help generate these maps. And so I just wanted to end on this slide, and this is generally where I start, but basically I have our agency and we support FDACs. We also work really hard to address the impacts of red tide. We've been kind of all hands on deck with this bloom, and it impacts other groups. We also have a dedicated manatee research team, turtle research team, and numerous fish and wildlife health teams that are working on this issue. And so I just wanted to stop and say thank you, because it involves a huge network of partners, and this doesn't even include all of them. So that's it. Thank you.
Wow, that was really good. I know I learned a few things. Thank you. Okay. Okay, so I'll change it up a little bit. We had some excellent science here, so now you should know everything there is to know about red tide and harmful algal blooms. And I'll go a little bit over what we do. We actually work with the good guys in the algae world. We're a company that was formed in 2006. We're located in Fort Myers, Florida. Fort Myers in specific is we're below the freeze line there, so we can cultivate algae year-round. We have over about 110 people on staff, with 50 people that have PhDs or advanced degrees in biology, chemistry, engineering. So we have a broad depth of knowledge in our company, not only about growing algae, but all the economics, all the equipment around growing algae. How do you separate algae from water? And doing techno-economic models on everything, because we're trying to make a profit from growing algae. We also, our main purpose is to basically produce natural products, sustainable products for algae for industrial and consumer applications. We have a large lab in Fort Myers. It's very well equipped. I'd probably say it's the finest algae facility in the world. I've never seen one anywhere near it. We have the personnel with the advanced degrees to operate this, and allows us to do a very advanced research on algae and all of the processing that we need to do to transform the algae into consumer products. We grow our systems and we grow our algae in enclosed systems called photobioreactors. These are basically large plastic bags that we hang on a trellis. And that increases the amount of light that the algae can get, and it's also an enclosed system, so it allows us to grow a specific type of algae at very high density. And to be economical in this, you really have to have a very high density culture. We have an extensive analytical chemistry capability. The reason this is important is we're able to not only study the physics around the algae and how they grow, but also we can get inside the cell and understand the biochemical pathways that are going on inside the algae, so we can understand the type of nutrient feeds that they need, also the exposure to carbon that they need to create biomass. And it also allows us to go in and say any algae that we bring into our plant, we make sure that it doesn't have toxins, so we can do a DNA analysis of the algae, and we can go in and understand through DNA sequence annotations whether these algae have the capability of making toxin. We are not interested in any algae that makes a toxin. To make money in growing algae, you really have to understand the process of doing it. There's a lot of inputs that go into growing algae. Growing algae is actually quite easy. Don't put bleach in your pool, and you'll have a bloom in no time. But growing a specific type of algae in very high densities is very difficult. And so you have to understand all of the economics that go around processing algae, to be able to make money on it. And so that's something that we have done is pretty much, if there's any technology that's out there in the world that you can use to separate algae or process algae, we have piloted it, understand the economics of it. And I think as we start talking about how to clean up some of these blooms that are out there, we need to bring expertise like that into play, because right now I think we're wasting a lot of money doing things that will never work from an economic standpoint. So a little bit, what are algae? You've got a good overview from our prior presentations, but I think the point here is this is a very diverse group of organisms. You can see down in the lower right, you've got algae, which is basically the slide that Dr. Rosen showed. But on the tree of life, you can see cyanobacteria are quite a ways away from most of the algae species. So we're really not dealing with one type of organism here. We're dealing with organisms that have very different places that they live and different means of survival. You can see that algae vary vastly in form. So when you think of an algae, I think most people think of little things floating around the water, but that's not actually true. Algae can take many forms. Here's some green alga. And you can see some brown alga. We've all seen this. Red algae. Dinoflagellates. This is the red tide that's happening out there. And diatoms. And then here's cyanobacteria. So as you can see that there's really not one type of organism. There are a whole class of organisms that take many different forms. And so when we group algae into a group, it's like grouping plants. And so you have to understand the differences between the two. And so, as I said, we're in the business of making algae, growing algae, and making products from it. And not all algae are bad. Dr. Rosen introduced a term to you called phycocyanin. Phycocyanin is, we actually grow a edible algae in very high densities, and we take that algae and we extract the blue from it, the phycocyanin, and that blue pigment is purified and is dried, and that is used as a food colorant. Synthetic food colorants are being phased out across the globe, and phycocyanin is actually the single best source of blue in nature. And so that's one of the products that we make. It's used in candies and all sorts of confectionaries. Other types of algae are used in cosmetics. If you look at the labels on some of your cosmetics that you use on a daily basis and you see an algae extract in there, it's very common. Hundreds of products have different algae extracts. The reason algae are used is algae are survivors. They've been around for a very long time, and they have built up ability to protect themselves from the environment. And then the things that they use to protect themselves can also be used to protect our skin. And so that's why they've been used in cosmetics for quite some time. And as you can see, there are lots of nutraceuticals that are used from algae. Many algae are called superfoods. But the use in nutraceuticals is quite broad, and what we're seeing here, especially in the PUFAs, the omega-3s, these are being done at an industrial scale and are being added into baby formula and all sorts of products. So not all algae are bad, and this is actually quite a growing industry. The thing about algae is the genetic diversity in the ocean is vastly greater than land plants. And relatively very few products are being pulled out of the marine environment and made into products for consumers. And this is changing. With the genetic diversity there are lots of problems that we can solve in our everyday lives using organisms like algae. So when we talk about HABs and red tide, this is a natural phenomenon. It occurs globally. We're not the only ones that have this problem. The exact causes really aren't very well understood, and it's certain that human activity is increasing the nutrient pollution that's going into our bodies of water. So that is definitely something that's in play here, but it's definitely not the only thing. As you heard before, we have lots of nutrients in the water, but we don't have a bloom. Why is that? And we really need to study this more intensively to understand this. to make sure we're working to solve the right problem. These organisms are everywhere. We will never get rid of them. They cannot be eliminated. And so we need to be working on what is causing the problem, not trying to eliminate the organisms that are out there. The extensive studies are needed. We need to given this type of problem this is on a global basis, very little study has been done about this, and this has to change if we're going to solve the problem. So what can we do about it? A lot of things are being done about it. I think that if you listen to the news media sometime, you think no one's done anything about it. But when you look at the comprehensive plans that they have for cleaning up water, I think that they're quite impressive. And we just need to have the funding and the political will to put the money into place and make it happen. The long time frames that we're having for implementing some of these plans in the 35 and 40 year, I think we need to rethink that timeline a little bit, because this problem is not going to go away. And the longer we wait, we could hit a tipping point where it has a much more dramatic effect on the state of Florida than what we're seeing right now. Local approaches are much more difficult to implement. The consumers themselves, the citizens of cities and counties, they need to have a lot more education thrown at them that they're probably part of the problem. No, they are part of the problem. We all are. And so we need to have more resources put at the local level in cities and counties, that the ones that are at the ground level dealing with this. So when there's a bloom and it's pushed up into a river like we have in Lee County, and people are pretty much driven from their homes, we're demanding action by our local, local politicians, and they're trying to do things that will never work. And so I think we need to have some type of means of giving state or the local governments the ability to look at some technologies that may or may not help and evaluate them, and not waste our money doing things that won't ever work. And I think the other thing that we need to do on the cleanup is introduce a profit motive here. As you can see, a company like us, there are ways to make money taking these polluted waters and converting them into proteins and other products. And so I think we need to also be thinking about how do we get a public and private partnerships going where there's a profit motive involved. And I think one of the biggest problems I see to solving this problem is the blame game. Everyone's pointing a finger at each other. The biggest boogeyman out there right now is agriculture and phosphate mining. But urbanization is a very big part of the problem. And so we think everyone needs to accept that we're all part of the problem, and then we all need to work together to solve it. It can't be solved by just focusing on one group of people. And so we need to go really change our behavior across the realm, which is a lot of education needs to be done at the local, state level. It needs to be almost like the tobacco commercials, where you're hearing it all the time, and I think we'll have some type of effect. A lot is being done, and a lot of things are already known to help solve this problem. We just need to start implementing that in a faster pace. So you can see that the best management practices, especially from agriculture, no farmer has ever made more money putting too much nutrients on his land. All farmers have basically a profit motive to not waste nutrients. And so it's really easy if we can help the farming community understand the best way to have best management practices, that we will see when we get the implementation that we're having much lower runoff. I think the urban areas are being let off the hook right now, and I think that's something that needs to change. We know we have data now that shows that a big part of the problem is the runoff from the urban areas. And that's the end. Thank you.
I think we have a few minutes for questions to try to stay on time. Are there any questions from the audience?
There's one.
Beth, in the back.
So my question has to do with this issue of the fact that we don't know much about what's causing
So we know, okay. Well, we know that the red tides start offshore. In terms of what causes them, we are interested in trying to get a better sense of the life cycle of the organism. We have some other types of algae, even some cyanobacteria, where we know that they form resting stages, and those resting stages almost act like seeds to help perpetuate the next bloom. We know that Carenia brevis is always present at low levels in the Gulf, but what we don't necessarily know is the causes, and I think having a better sense of the life cycle dynamics would be really important. That automated microscopy technology that I showed you is one of the tools that we're trying to use to get a better handle on that. We're also trying to build better instrumentation in the lab to help simulate the environment so that we can use cultures of these algae similar to what was shown, and to try to test different things, examine how different parameters co-vary and may cause red tide to do different things. And so right now, for example, using LEDs, we're able to simulate surface light levels in the lab. Previously, all of the experiments, or a lot of the experiments that had been done, had been using much lower light levels. So those are some of the advances that we're using to continue to learn more about the causes and really the ecology of this organism.
Any other questions? Diane?
Okay.
Can they have
Diane,
microphones?
can't hear you.
Yeah, could you speak up, please, ma'am?
I read a article earlier this week about air quality monitoring, and you're going to be going through beaches every 3 hours, whether they're closed or open. Can you speak to that?
So the Department of Health has recently initiated a program where they will be doing some additional respiratory irritation testing. This is building upon a prior project where they were able to document that the aerosols can make it up to 1 kilometer inshore. This will be a new study, again, led by the Department of Health. Moat Marine Lab will be involved. They're still picking sentinel sites and setting up the experimental design, but that's something that is evolving rapidly right now.
There was another here, second. Go ahead.
Yeah, good morning. Thank you for the time and presentations. My question is regarding any research or technologies that we have available now for immediate evaluation of blooms and identified. For example, I know Moat Marine has been doing some research with ozone, kind of on a localized level now. Can you speak to that a little bit?
Yes. Unfortunately, what you're dealing with, these algae, although they look very dense, they're actually not. So when you try to separate them from water, you're talking about a very expensive proposition. Ozone, we can tell you for sure, we've piloted ozone. Ozone will never work. It's going to be way too expensive. Ozone is generated by electricity, and then the amount of ozone that's needed to actually sterilize the water itself is based on the bioload of the water. And so you can imagine trying to take water from the ocean and how much bioload that's going to have. We're really not talking about an option here. So there aren't a lot of technologies that are available here. We need to have some innovation around this, and we need to bring some real techno-economic modeling around these types of things. A lot of the cities and counties that we can see that some of the things that they're doing are completely wasting their money. They'll never work. So unfortunately, there's no silver bullet, and there's not going to be a white knight that rides in and kills this problem with one thing. It's going to take a lot of different things to solve the problem. Sorry.
Okay, well, I guess at the end of the day, we're going to need to bring it in for a landing. They told us we're going to be out of time here very, very quickly. So if you have any additional questions, I know there were some hands that came up. I'm sure these folks will be around, would happy to share. One quick point. If we continue to invest in research, I think you can see we've come a long way in understanding this challenge and what mitigation may be available for us. And every time you open one door in research, it may open up 5 more, right? So if we're diligent and we continue to stay focused on funding this research, I believe my glass is half full, but I believe in short order we're going to understand what real mitigation techniques are available or what we can make available and begin to stem the economic challenges that come from these, these algal issues. Anyhow, thank you guys very much. Have a great day.